I want to welcome two of my favorite people. Jennifer Ferguson, she's a family nurse practitioner at UCSF Benioff Children's Hospital in Oakland. And she's been working on the Deferiprone project trial since 2014. So she's been in contact with many NBIA families and also with families with other rare disorders like neurological and hematological disorders. And she is our go-to person every time we need a question or we need some sort of help, any doubt, we just shoot her an email and she's on it right away. We know we can count on her. And then I want to welcome also Dr. Vichinsky. He says that the healing arts is a calling for him and it's a way for him to make a difference in the world. And he certainly has made a difference in the world of our family because we were desperate back in 2007. We heard that Dr. Vichinsky was using Deferiprone with other disorders, and we approached him to see if he could help us out to try it out with Brent. And he didn't know anything about PCAN or NBIA, but within a week we had an appointment. He had talked and got caught up with a neurologist on what was PCAN about. And he went to bat for us for the FDA and got us the compassionate use. And that has made a huge difference in Brent's life. I think he has stabilized him and he regained certain skills he had lost. So we are forever grateful to Dr. Vichinsky and his team. So please, let's welcome them to the stage. Thank you.
Thanks, everyone. I just wanted to do a quick introduction by way of what Veronica said, which when I joined Dr. Vichinsky about 5 years ago, he was already well on the path of starting this study. And I heard the story of Veronica's advocacy for her son and how Dr. Vichinsky stepped up and was able to make the access to the medication happen for him as well as other patients, and then how it turned into this big multinational trial. And it just really struck me that a single parent or family and one or two providers can make such a huge difference, especially in a rare disease like this. So patient advocacy is just huge in this disease, and I think we've seen that throughout the conference, how much difference one or two people can make, and then the power of all 250 of us who are here, how much potential we have to make change. So that's been a wonderful thing for me to see as I've joined this community over the last 5 years. So we're here today to present the results of our Deferiprone trial in patients with PCAN. This was an 18-month random controlled trial, placebo-controlled trial with patients with PCAN from ages 4 to adult. And after the first 18 months, everyone rolled on to an open-label extension trial where they were all getting the drug. So I'll let Dr. Vichinsky present the results, and then we'll have plenty of time for questions at the end.
I'm delighted to be here, and thank you. You know, if you have a rare disease, one of the only ways you're going to change things is by being an advocate. And so I encourage you really to lobby politically and socially with the FDA or the government, because someone who deals with multiple diseases, it isn't necessarily how common it is. It's really where you put your political punch. So I think Brent's family started their political punch on Deferiprone with me. First of all, they're a wonderful family, and he's a handsome guy. But the real reason is he makes incredible gelato. So he makes the best pistachio gelato, and I'm here forever to get that. So let me go ahead. So basically, I started treating Brent and some other patients on a compassionate use trial, and after I had 8 or 10 patients on it, using an IND, the FDA said, what do you just can't do this and keep enrolling people on a compassionate use. We're closing you down. You have to do a trial. And I didn't have any money. Eventually, I got a grant, or an exploratory one, to start the trial, a small trial, and Susan Hayflick and Penny were part of it. And just when the smaller trial was going to start, the international organization for NBIA approached, asking if the trial we had could be enveloped into this international organization as a phase 3 trial. So all of us linked together, and with many investigators, as you could see in this picture, Thomas Klumstock and Newcastle, and a number of other, Italy and other places. The study became so big in an attempt with a rare disease to prove the drug has benefits so it could be released. We approached Apple Pharma, who made Deferiprone, to help us, because we really didn't have the infrastructure to do the sophisticated statistical analysis and FDA monitoring, and thankfully they did do that. So as you know, PCAN is a rare disease. It's the most common type of the NBIA. There are probably 2, maybe 3 cases per million. It has a protean amount of symptoms. Dystonia and Parkinson-like movements are only one of them. But one of the things that characterizes the disease is this large amount of iron in the brain in a particular area called the globus pallidus and substantia nigra. This is a very critical part of the brain that sort of acts as the electrical station for other parts of the brain to communicate. And while the other parts of the brain may be totally normal, they don't function because they have to communicate through this area. And dopamine and the other enzymes in this area are basically very rich in iron. And when the body's trying to make the coenzyme A that's missing in PCAM, it inadvertently has to use, keeps using up and releasing iron into the tissue of the cell that leads to a lot of destruction. The reality of it is there is no effective therapy for PCAM. It's palliative. Some people have used DBSs, baclofen, Artane, but most of the therapy is really supportive nutrition, and exceptionally important is physical therapy and occupational therapy. This is the first time ever, really, there's a randomized trial to see prospectively whether there's a therapeutic agent that could change the natural history of the disease. And this study is an attempt to prove that. Here is the picture of the brain, and if you cut it in half and you're looking down, and you could see the yellow there, and that whole white area under the yellow is iron, and it's a chunk of iron that doesn't belong there, and the blue is an area of a different color without anything in it, and so it creates this, what's called the eye of the tiger. But it's that white part that is a chunk of iron that gets deposited there that sort of creates havoc in the attempts of both sides of the brain to communicate and function. So this study is trying to see if we have a new type of therapy. The drug Deferiprone is an important oral chelator. It's been around, and it's unique in that it's a small molecule and doesn't have a charge, and it can easily enter into the brain. The brain has what's called the blood-brain barrier, and it doesn't let drugs into the brain easily. So the other drugs don't enter the brain, and Deferiprone does. So it's a natural chelator to detect. We also have data. First, we now can quantitate iron in the brain exactly, and we know from other trials that that you can remove this iron with a chelator. So why not just give it to everybody? You really have to prove a drug works. And every drug, there's no good and bad drugs, in my mind, as an oncologist. It's really a ratio of the risk of the drug to its benefit. So the risk of this drug is a idiopathic sudden lowering of your white cells, the cells that fight off infection. You don't know when this happens. It's always reversible, but if you have a low white count and you don't know it, and you got a blood infection, the germs would grow in your blood and could kill you. Now, if you have a low white count, it's always reversible, but you have to be checked if you have a fever particularly, so you can be seen and treated. So it's important to have your white cells monitored. And that's a major burden with this disease, was the weekly CBCs needed. So the study was started really to see if reducing brain iron will happen and could it improve the disease. This is just a kind of cartoon to let you know that iron is really dangerous when it's in excess. And as you know, mitochondria are the energy-building blocks where a lot of important enzymes and proteins are made. And iron accumulates in there in PCAM and destroys a lot of its function. You know, if you've ever had a back of a car that rusts, rust is iron that gets oxidized. And really, that's what we see. That's the same analogy within the cell. The iron gets into the cell, and it basically rusts the membrane. So the lipids, your membranes of cells are made up of lipids, and the lipid gets dissolved, and that's called peroxidation. And you're all familiar, if you actually iron oxidized DNA, you can destroy important parts of proteins and DNA structure. And you're all familiar with everyone always talking about, did you take your antioxidants? Well, the common pathway of injury is oxidation, which is largely driven through iron as its mechanism. What we know is deferiprone can get into the cell, and it can lower these oxidative materials called reactive oxygen species. So the thing we know from other diseases is that deferiprone can lower the dangerous components that happen when you have too much iron and what dissolves the function within the cell. And we know that in PCAM, there's too much iron in the mitochondria, within the cell, and within the surrounding area. And so we can now see if giving this drug has any benefit to the patients. And I basically summarize this to you. It's a drug that it crosses lipid membranes. It easily enters into the brain and gets into the mitochondria and the organelles where a lot of the important enzyme work is for PCAM. And it lowers ROSs, which are oxidants. So this large study was designed, and including many countries, as you see here, and it was a randomized, double-blinded, placebo-controlled trial. There were 90 people enrolled on the trial. They all had to have the definition. They all had to have PCAM confirmed genetically. And there were ethical issues about doing this, and the way we decided to make it fair and still answer the question was, and because the FDA really wanted a randomized, placebo-blinded trial, what we did was weight the study so that for every patient who got placebo for 18 months, 2 got the drug. But as an answer for everyone, after 18 months, everyone was put on the drug for another 18 months. So the study went on for 36 months. The first 18 months was a placebo-matched, a treated group, and the second 18 months was everyone was on the drug, and we followed how they did. And this is important because the benefit of a drug is important to evaluate over time. How powerful the drug is in part delivered by statistics, but also duration of exposure. One of the big problems with PCAM, as you know, is how do you determine they're getting worse? And the FDA really wrestled with this, and we did. And it was eventually decided that we would use a number of endpoints to try to figure out if the drug was helping or not. The Barry-Albright dystonia scale was used, and this is the classic scale. The higher the number, the worse, the more dystonia. That is used for Parkinson's, and it was chosen. It's on a problem in that patients with PCAN can deteriorate, and the bad may not detect that, or vice versa, but it is globally coordinating with the progress of the disease. And there was a bunch of other important secondary and other outcomes, including did the iron go down, were there motor abnormalities or motion abnormalities in the extremities. The WEFAM looked at things like cognitive function, quality, and then there were separate scores on how the family rated quality of life. So there were 60 patients in the treatment arm, 30 in the placebo. Everybody anyone was eligible, if they were over 4 years of age, had to have PCAM. If they had a deep brain stimulator or they were on Baclofen pump, all these things were eligible as long as it didn't just happen in 2 months before. And the exclusion criteria were obvious in that if they were taking Deferiprone already or had overt toxicity to that drug or other blood problems that were not related to PCAM. And so I told you the study was divided in first the placebo controlled arm for 18 months, and then another 18 months where the same patients were tracked. This just summarizes who these patients were, and it gives you an idea. And it is important when you start seeing what made a difference. They were older overall. They were 19 and 21 year of age. Basically, the age of onset in both groups was 7.5 years and 8.4 years. And most of these patients had the disease a long time. The placebo arm had a 12.7 years, and the Deferiprone arm had a 13 years. And the sex ratios were relatively similar. Most patients were Caucasian, majority anyway. And they had to be eligible, you had to have a significantly abnormal bad score, and both groups were abnormal at 16.5. The Deferiprone was somewhat worse. And both groups, the MRI R2 is a measurement of brain iron. Both of them had severe amount of brain iron on entry. Well, what we were so impressed with, and these initially, was those who stayed on the trial, they took the drug. We were really impressed. This is a community that's highly motivated. There was enormous compliance with the study, best study I've ever been in terms of patient compliance. And the other thing that we noted right away, and is what the primary method of operation of this therapy was could the drug remove your brain iron? So here, if you look at this, these are the far left one, that square, is what's called a region of interest. That's where the brain iron was quantitated in milligrams. And if you go all the way over to the right, where there's a histogram, the red line is the placebo arm, and you could see from 0 to 18 months, the placebo, the amount of brain iron never changed, right? But in the Deferiprone treated group, you could see the blue line that's dotted goes straight down during the 18 months. So within even the short period of time, there was a marked benefit of the drug once it was started in removing brain iron. So while I have a lot more data, I could tell you the drug removed brain iron. But remember, these patients were treated, had this disease a long time. One of the things I was interested in as a clinician is what clinical benefit did the drug seem to have to me when I was following patients. Well, one way to determine this was how many of the patients required breakthrough medications, which means beyond the standard meds they were started on, how many had to have added rescue meds during the therapy. Ideally, the NIH, FDA would have liked everyone to say, you're on these meds, nothing could change, you have to stay on it, but it was clear that wasn't going to be the case. So we tracked on the study a rescue medication in each arm. And what we saw here was that in the placebo group, 21% of the patients required rescue treatment for worsening acute symptoms, and 11% of the Deferiprone arm. Now I'm going to show you some slides, and I'll try to explain them to you in several ways. This was, if you an intent to treat study. So what that means was, once you entered in a study, you're on the study. If it turns out you dropped out of the study, or you stopped taking the med, you stayed in the trial. That's what an intent to treat is. So this is a report of all the patients in the first 18 months of the study, comparing placebo to Deferiprone, and everybody, whether they dropped out or not. And you could see the trends here. In other words, red is placebo. The higher the number, the worse you do on your bad score or your dystonia, and blue is the drug. And what we see is a consistent deterioration in both groups over time, but it's descriptively, it was consistently worse in the placebo group. In this analysis, which everyone lumped together, the trend is very clear, but they did not reach strong statistical significance, probably because of the intent to treat design, and I'll show you what I mean. When we analyzed the groups and we took the atypical patients in just the first 18 months, remember, atypical really means these were the older patients. The groups were stratified by age, and PCAN is pretty well stratified by young kids, young and older ones. This group showed a strong statistical benefit to the drug. And you could see in each age group, each 6-month period, the atypical patients had a statistical improvement, did statistically better than the control arm. Both groups were getting worse, but it was the decline was much lower in the Deferiprone treated groups. So this was a statistically meaningful in every period it was tested. In contrast, in the severe young children, we saw the same trend happening over time, but it wasn't statistically significant. These children do worse, as you know, and I'll go over with you later on why this is the case and what I think are the variables. But they did do better overall in the Deferiprone group, but the magnitude of change did not reach statistical importance. So what we wanted to do was really move into the second half of the trial, which was the second 18 months. And to really get around the intent to treat and all these other issues, what this data I show you now is everyone who finished the first group and went on to the second half were analyzed. So that way we knew everyone who entered into this did the first half of the trial, and we could look at that. So this is an important slide. This is the first 18 months of those patients who finished the full study in the first 18 months and went on to the second half. And what we saw here, remember, red is the placebo and blue is the Deferiprone, and the magnitude of change, the column is how much worse the bad score got over these 18 months. And there was a statistically significant benefit to the drug in this when you compared them of those who completed the first study. So this was important to us and important clinically. Let me see if I could. If you looked at the Deferiprone group, let me go back here. I think I messed this up. Yeah, so now this slide then looks at, we took the children and adults who were on the placebo arm the first 18 months, and then for the second 18 months got Deferiprone, all right? So this was really important because we have their 18-month data from being placebo, and then they got the drug. And what we saw was a significant difference between how these patients who were their own control deteriorated when they're on placebo versus the drug. So you could see that during the period they were on the drug, the decline in the bad score was much lower than when they were on placebo. And yet the Deferiprone groups never changed. In the first 18 months to the second 18 months, or 36 months, they retained the same steady state lower decline. I'll skip this, but this is just a statistical interpretation of the same data I showed you. Basically, the first two are, again, just showing you that at the patients who had placebo in the red, and then on the second blue mark is what happened to them when they went on the drug, and showing that their magnitude of decline was markedly lower, and it was statistically important. Another way to analyze how patients do when they have all these symptoms is what's called a forest plot. And a forest plot is basically you list in categorical method, like an outline, all the tests you do, and then you put a 0 in the middle, and if there's data to suggest the drug is helping, then you score them to the left. And if you think the data suggests the drug is not helping, and then you score it to the right. And this is a common used test. So what this shows is most of the tests we did, regardless how statistically strong it was, 19 out of 24 of all the tests were always in favor of using the drug, which is important to me in that it shows it isn't just an isolated event, that there's that all the parameters showed some degree of some effect, except for a few. There are a few things here I'll show you that I think need to be at least mentioned. The ones that really stood out in this forest plot were really the dystonia of the mouth and the lower extremity, the right and the left leg score. It seemed, in contrast to the placebo, that those three tests that the drug appeared to have benefit. And the other thing that it had benefit on was in the WeFIM cognitive functioning. And what we found was that cognitive testing, or memory recall, was, it seems the drug improved that. And you could see that if I had a point, maybe there is a pointer here. I don't know. Terrible. But anyway, you could see that. So memory, cognitive function, lower extremity dystonia, neck seemed to really stand out in this analysis. So we went back in this forest plot to try to figure out what are the variables in the different, why do some people get better and some not? Well, what we found here again is the middle is the 0, and that getting better is to the left. But we found here is the bottom line was duration of the disease. So if you had the disease symptoms for under 5 years of age, you did much better. And so what this suggests to me, if you can start the drug early enough, closest to the time of diagnosis, so that the iron isn't sitting in there causing damage, you may respond better. And so the earlier you could start the drug to the time of diagnosis, and this makes sense. This is why newborns who are born with hypothyroidism are immediately started on thyroid. You don't wait till they get symptoms and so forth. The other thing that seemed to make a difference was the age of onset. Older patients over 6 years of age seem to do better than those younger than 6 years of age. And they may be accumulating iron at a slower rate than the younger group. And therefore, this is my interpretation, that the younger group, it underscores the need to start their drug as early as you can. So then we looked at adverse events, and there are really only two things that are worth commenting here. First, there was anemia, or in the Deferiprone treated group, no one needed transfusion, no one had severe anemia. The anemia was induced by the drug that caused iron deficiency, and it was totally correctable by iron. We analyzed the data in detail to see if giving iron would make the disease worse or better, and it didn't have any effect other than correct the anemia, because the excess brain iron is internally driven, not from what you take. And the other thing that was interesting to us and me is at the very end of the slide, you could see freezing phenomena, and that while the numbers were small, 10% of the placebo group had serious freezing events, and none of the Deferiprone group had serious freezing events, suggesting that Deferiprone may have a protective effect. So if one summarizes from my standpoint, where I saw this as a clinician, is it does appear the drug slows the disease progression. And while it is not statistically significant in all time periods, overall it is, and that the drug was well tolerated and the safety profile was very good. The things that complicate and that stand out are the disease duration. If you're young and you had the disease without treatment for a long time, the drug may have less of an effect. And the age of onset may be a variable. Older patients may do better. We did noted a difference in rescue meds between the placebo and the drug treatment group. We saw encouraging data on cognitive and memory, and encouraging data on some of the bad tests consistently, particularly dystonia in the lower extremity, and some benefit in freezing. After the patients finished the second half of the trial, which was the open label, they had the option to go into a compassionate use trial, in which many have chosen to be on compassionate use. And what the company was able to work out with the FDA, which was quite exciting to me, was they allowing this group to only have CBCs once a month. And so far, that trial's going along, and there have been no, to my knowledge, adverse events with monthly. What the long-term release of the drugs, FDA guidelines, will be determined, not by me, but that is. So what do we want to do? Well, this whole study I presented to you was a group effort. Really, first of all, it's really the patients who made this and the families made this happen. So what we want to do is share this with the medical community and the stakeholders, and then we share it with the regulatory agencies, and with the goal to get it approved. And that's what we're trying to do now. Personally, depending on how you feel with this data and your experience, someone who's been through the FDA for rare diseases, this is the community groups have a strong voice in rare diseases and advocacy for this drug would carry a lot of weight if you wanted to. The FDA is responsive to that. In my experience with other drugs, XJ being one, in which it did not meet its primary endpoint, but it met a sub-endpoint. The drug got approved. And a lot of these, so I'm encouraging you, if you're interested in getting this released, to advocate for it, or whatever drug in this disease you want to advocate for by calling or writing to the FDA. And that's really it. And I'm open to any questions if I didn't totally confuse you.
Thank you so much for the presentation. It's absolutely wonderful work. A question for you regarding the iron accumulation that you showed. Very interesting results, and the placebo group did not appear to have a change in the amount of iron in the globus pallidus. Maybe equally interesting is it did not seem to go up. So can you comment on whether you looked at iron levels in different age groups, classical versus atypical, and whether there were any groups that you saw actual increases in iron load?
I don't have that. I can't. I think I have. My impression is the accumulation changes over time, and it changes with mutation. But I don't have the hard data to give you that exact answer. Maybe Fernando does from looking at it. One of the problems is we did not mandate that everyone had to have an MRI done, and so there is a concern in fragile patients putting them to sleep or doing it that you may destabilize them. So the MRI data was biased in the sense of who would go through it. And the most unstable patients were ones we didn't do. So that's all I could tell you.
I'd like to ask a question. If the trial was successful, which it sounds like you're saying, why doesn't the FDA just approve it?
Well, I'd like them to do that. I'm a clinician scientist, so someone has to take the case to the FDA. Apple Pharma does, and then what they have to do is present the data, and then they review the data, and then the FDA makes a decision. And many drugs, I've been through one now recently, is they get what's called conditional approval. They approve it in the short term with the goals that you would finish the definitive part. And in this trial, that may be necessary because the primary endpoint of this trial was to show a statistical significance in the entire group over time. And though we saw it in subgroups and different periods, the whole entity of the data wasn't presented yet. So I imagine that the FDA is going to review this soon, and they will be, and I'm not there at that table, but they will be discussing on what part and what questions. But I think, and that's all I could tell you because I'm not part of that team.
Two questions. Am I right? You said they did a weekly blood test?
Yeah, in this study, which was quite a tour de force, frankly. I mean, it's impressive to me that all the patients did this. So we set up a central system where the patients went to Quest or were everywhere locally, and they had to have a blood test to see if their white cells were low, and the labs were sent to us directly in California. And that is a requirement of the FDA to make sure that no one's white count is going down. The study did not find that problem happen in this group, but there were only, let's say, 100 patients. If there were 1,000 patients, there would have been a case or two. So what this study did show, and which was the concern, is this group did not appear to have any additional risk of taking this drug. In fact, the rate of there were no cases, so it was very low. There was a concern, would it be higher in people who don't have iron in their body? Some people thought the drug would bind to iron and it would cause neutropenia when all the iron was out of the body. So this is a landmark in that case, is that the drug seems safe in non-iron overloaded patients who have just local iron. There's a hope that, and there's some information to support it, that the weekly CBCs aren't necessary. A lot of that data stems from some cases that happened in Europe or India where the drug was given out, and not for this disease, but no one ever had their labs checked, and a few children got sick, never got treated, and a few died.
And dumb question, can white blood cells not be done by a finger prick at home?
Yeah.
You have to go to a lab?
No, I think you can. No, I think white cells can be done anywhere. Home or bath, wherever you want it. It's just a finger prick.
So that was just an FDA requirement. Okay, thank you.
I'm hoping that will change to once a month.
My daughter has BPAN, And so we took her to a neuro-ophthalmologist, and he said she had iron accumulation on the optic nerve, which has caused partial blindness, peripheral blindness in the eye. And he said it's not a matter of if, but when that she will lose her vision. Does this drug only pull iron from the brain, or is it more like a total body chelating agent?
Well, I could tell you this. There's no good trials for BPAN, though there's an interest in it because they have excess brain iron. And I am treating under compassionate use a number of, shall we say, desperate or very ill children with mutations like that because there are no other options.
And one more question is, I know some chelating agents will strip the body of certain vitamins and minerals,
Yeah.
and they have to be doped in order to maintain a healthy level. Does this drug have any potential, or does it?
Well, yeah, I think this drug has it's a fairly specific drug for iron, but it can make you iron deficient, and it can lower your zinc levels. And so we do monitor zinc levels in the patients periodically. A few others, but mainly zinc.